Good evening, Arkadyut. How are you? Good evening, good evening. How are you, Arkadyut? Okay. Okay, let's continue. Good evening, all. Good evening, Pankaj. Okay. Okay, anyone else is going to join? Okay, let's wait for 30 seconds and we'll start. Good to know. Good evening, Savita. Good evening. Okay, so in the yesterday class we couldn't do the class, so that prokaryotic replication and eukaryotic replication I will be providing you in the form of a recorded. Okay, so you can watch the recorded. I'll upload tonight. You can watch this. So today originally the process of transcription has to be continued, so we are going to continue the transcription. So once again I'm repeating the process of prokaryotic replication and eukaryotic replication video. I'll be provide you in the form of a recording. You can watch that. Okay, now let's start the process of transcription. Okay, so friends, every one of you has to understand where does this particular transcription occur, and first of all, what do you mean by the word transcription? Okay, so friends, we read the central dogma. That is what the DNA is the one which. Good evening, Saima. The DNA is the one which is basically involved in the synthesis of a protein. Good evening, Akshay. Please continue. Please continue, everyone. It is involved in what, friends? Synthesis of the protein. Okay, I'm just saying to everyone, the prokaryotic and eukaryotic replication part I will be providing you in the form of a recording. I'm requesting all of you watch that without fail. Since today is the 4th day, so we are going to continue the transcription as it is. Okay, hope it's clear for everyone. Yes. Okay, perfect. Good evening, Trina. Good evening, all. Okay, now friends, listen. The process of transcription. We all know very well, friends. I told you many a times what is the ultimate goal of the DNA. Ultimate goal of the DNA is what, friends? Synthesis of a protein. But friends, we all know very well that even though DNA having the ability or capability to synthesize the protein, DNA never ever involved in the synthesis of protein directly. Instead of involving in the synthesis of a protein directly, what the DNA is going to do, friends? DNA is going to give rise to an intermediate molecule. What do we call that particular intermediate molecule, friends? We call that intermediate molecule as a RNA. Now friends, that particular RNA undergo the process of translation going to give rise to the protein. So friends, everyone quickly answer my question. My question is, if I want to synthesize the RNA from the DNA, do you think I require the entire DNA, or I require only a part of the DNA which is involved in the translation? Do I require entire part of DNA, or I require a part of the DNA? So friends, everyone listen, this is an entire DNA, right? Friends, whenever there is a involvement in the synthesis of protein, everyone listen, friends, we will take only a little part of the DNA, is it? A little part of the DNA is required to synthesize RNA. Everyone listen, only a part of the DNA can be giving rise to what, friends? RNA. Never ever forget that point at all. And friends, where is the process of transcription is taking place actually? We all know very well, friends, in case of a prokaryotic organism, the process of transcription is exclusively taking place in the cytoplasm. In case of a eukaryotic organism, the process of transcription exclusively taking place where, friends? In the nucleus. We all know that very well. So listen everyone, look into the definition of a transcription. Transcription is the process by which the information in a strand of DNA. not the entire information, friends. I can use a word, a segment of the DNA. A segment of the DNA is copied into a new molecule of RNA. What do we call that particular new molecule only? We call that particular new molecule only mRNA. Friends, everyone understand how many types of RNAs are basically synthesized in the process of transcription? We all know that, friends, mRNA, tRNA, rRNA. Okay, I have asked you many a times a question also. Among all the types of the RNAs, we all know, friends, rRNA is said to be a most abundant form of the RNA in the animal kingdom, which is rRNA is the most abundant form. So friends, we know that whether it is a mRNA, whether it is a tRNA, whether it is a rRNA, doesn't matter, friends. All the three type of RNAs are extremely required, extremely involved in the synthesis of protein. Friends, even though all are involved in the synthesis of protein, their site of synthesis is entirely different. Friends, anyone can quickly tell me where is the site of production of rRNA? Anyone? Where is the site of production of rRNA? Okay, there's no problem. We know that the site of production of rRNA, where, friends, it is in the nucleolus. Is it? And friends, what about the mRNA and tRNA? Friends, both mRNA as well as the tRNA, both are going to be synthesized where, friends? In the nucleoplasm. Friends, please kindly don't forget, okay? mRNA, tRNA synthesized in the nucleoplasm, whereas, friends, rRNA is basically synthesized where? In the nucleolus. Kindly do not forget this particular point at all. So friends, everyone listen, the name itself indicating, right, mRNA. What do you mean by that? Messenger RNA, which means which exactly bringing, please try to understand, which exactly bringing the information from the DNA in the form of nucleotides. In the form of nucleotides. That is what the function of mRNA. So friends, it is actually bringing the information from the DNA in the form of a nucleotides. That's why we call it as a mRNA. It carries the protein blueprint from a cell's DNA to its ribosome, which are the missions that drive the protein synthesis. Next one, friends, tRNA. What did you understand by the word tRNA, friends? tRNA nothing but it's a kind of a RNA molecule. Everyone has to remember, my friends. Whenever I say tRNA, I want every one of you remember one statement. What is that, friends? tRNA nothing but, friends, adapter RNA. So what it is adapting actually, friends, it is adapting the amino acid, because without combining amino acid there is no way we cannot synthesize the protein at all, is it? So now what is going to happen actually, all the amino acid come close together with the help of a condensation reaction, they're gonna form the polypeptide bond. But the point is very clear, my friends. How does this particular tRNA will enter, how does the tRNA can bring the amino acid? Friends, I want every one of you remember this particular statement, either for a part B or a part C or a generalized statement. You never ever call, good evening Deepika, you never ever call tRNA is a charged tRNA until and unless it is associated with the amino acid. If a tRNA is not associated with an amino acid, I can directly call tRNA is a uncharged tRNA. Never ever forget this particular statement at all, friends. So you people listen, tRNA is what? Charged tRNA whenever it is encountered with the amino acid. Anyway, we will read in the translation process easily. What is its function? Just now I told you that it is involved in the adaptation. What it is going to adapt? It is going to adapt the amino acid. Now, among all, the most important rRNA. This is the one. What the rRNA will do, friends? We all know very well what the rRNA doing. Whatever the information is there, whatever the information that is coming from the mRNA and amino acid, by joining the information with the amino acid, rRNA is actually involved in the synthesis of protein. That is the unique function of all three RNAs. So what is the point every one of you has to remember, friends? Either directly or indirectly, all three type of RNAs are exclusively required for the synthesis of protein. Never ever forget that particular point at all. Now friends, now it is must and should. We have to know the major fundamental differences between the DNA replication as well as, friends, the process of transcription. Now you can ask me, sir, is there any necessity that we have to read the differences? Yes, friends, you have to read the differences. It is compulsory. Right. Now I will tell you very simple differences. So friends, first, except these or apart from these also, let me tell you another point. So when I say replication, you all know very well, friends, replication means whole genome get copied. Copies of a whole genome. But when you heard the name of a transcription, what you have to understand, only a part of the DNA is getting or giving rise to the RNA. Don't forget, we all know very well, friends, DNA replication special, where the DNA replication is going to occur, our cell going to prepare for the DNA replication in the G1 phase. After completion of their preparation, the DNA is going to enter into the S phase of the cell cycle. We all know, friends, S phase of the cell cycle, we will be calling it as what, friends? The phase where the DNA replication occur. Everyone keep it in your mind, friends, where the transcription is going to be taking place. Transcription, friends, exclusively taking place in the G1 and G2 phase of the cell cycle. Please kindly don't forget these statements at all. And friends, we read in the rules of replication very clearly. What is that we read in the rules of replication? It requires RNA primer for the replication to start. What do you mean by that? What exactly you understand? We read many a times our DNA polymerase enzyme cannot start the process of a replication without a primer. So that's why DNA polymerase enzyme always dependent on the primer to initiate the process of replication. What about the transcription, friends? Transcription is the one which does not require primer. So every one of you has to remember one of the most unique point here is that, friends, you know, this particular RNA polymerase is a versatile enzyme. It do not require any primer. Actually, friends, the RNA polymerase enzyme having intrinsic primase activity. Please kindly do not forget this particular point at all. What, friends? RNA polymerase having intrinsic primase activity. That's why it does not require a primer. Please kindly don't forget this particular statement at all. Now, what are the major enzymes that are involved here? DNA polymerase, DNA helicase. Now, friends, everyone has to remember my statement, whatever I am talking. Is there any necessity of the helicase in the process of transcription? No. Now you can ask me a doubt, sir, if there is a no necessity of the helicase in the transcription, then how double-stranded DNA is going to get separated? Friends, the double-stranded DNA gets separated by the RNA polymerase itself. Why? What is the reason? The reason behind that, RNA polymerase enzyme having intrinsic helicase activity. Do not forget that point at all. What it is having? It is having a intrinsic helicase activity. Without any extra helicase enzyme, RNA polymerase only can open the double-stranded DNA. I'll show you in the upcoming slides. As I told you, entire copy got genomed, entire genome got copied, only a certain copy, or a certain part of the gene got copied, and we commonly know that what kind of nucleotides can be involved. So friends, this is the one of the most important reason important difference you have to remember. What is that? I was continuously saying that DNA never ever involved in the synthesis of a protein directly. Shalini, RNA polymerase having intrinsic helicase activity. What do you mean by the word intrinsic helicase activity? Intrinsic helicase activity in the sense it is going to or it work like helicase. It do not require any extra helicase. What is the function of a helicase? Open the double-stranded DNA. RNA polymerase intrinsically having the helicase activity, hence it only can open the double-stranded DNA without the help of external helicase. You got it? Yes. Now friends, this is the most important statement every one of you has to remember. As we are saying that the DNA polymerase enzyme, DNA not involved in the synthesis of protein directly, even though it is having the ability or capability. My friends, one of the major reason for this is, everyone listen to me carefully, conserving genomes for the further generation. This is the major reason. Means it has to store the information for the further generation. Now you only try to understand. Suppose example friends, if the DNA starts involving in the synthesis of protein directly, there is because of certain mutation DNA got damaged, it become permanent mutation, it passes to the next generation, and the cancer, it leads to many genetic abnormalities, even cancer also. So that's why to protect its stability, to conserve its stability, the DNA never ever involved in the synthesis of protein directly. Only it can give rise to the RNA. Never ever forget that. Which activity, Lakshmi? Aishwarya, no, whether it is a prokaryotic organism or eukaryotic organism doesn't matter. RNA polymerase enzyme having the intrinsic helicase activity. In prokaryotes also, eukaryotes also, it does not require external helicase enzyme at all. And we all know, right friends, everyone look into the picture whatever I am drawing here. Suppose example friends, whenever the DNA replication occur, this is one parent strand, this is another parent strand, and what we gonna get, this is one daughter strand and this is one daughter strand. Now you only tell me, whenever there is a replication occur, do you think after the replication, do you think parent strand and daughter strand will be separated? No, not at all. Daughter strand and parent strands cannot be separated. They will be with each other only. But friends, now you try to look into the transcription. So this is the DNA. Once the transcription occur, what is going to happen friends? The mRNA comes out. mRNA comes out easily. So in the DNA replication, the daughter strands will be associated with the parental strand, but in case of a transcription, mRNA completely comes out. Now here comes a very unique point in terms of the degradation. You know friends, products do not degrade. You know, I am not saying that the DNA cannot be degraded 100%. No, I cannot say that kind of statement. What I am saying is that friends, when compared to the RNA molecule, the DNA is going to be degraded at a very less amount when compared to the RNA. And RNAs can be degraded very faster when compared to the DNA because of the action of many vulnerable enzymes. We're gonna read that in the upcoming slide. So friends, these are all what? Common fundamental differences between the process of replication and the process of transcription. Friends, quickly tell me, is that clear for everyone? So far it's clear? Just say yes or no. Is it clear? Yes. Okay. Perfect. Okay. So friends, now let's try to understand how does the process of transcription is basically taking place. Friends, before understanding the process of transcription, I want every one of you has to understand one important line about this particular transcription, especially in the prokaryotes as well as in the eukaryotics also. What is that? That is what do we call polycistronic, then friends followed by the monocistronic. It is a very important, you people has to remember this. What is polycistronic property as well as the monocistronic property. Okay friends, everyone listen this. If you look into the mRNA, friends, this is the mRNA. We are talking about the prokaryotic organism. In which direction mRNA can be synthesized? We all know that friends, mRNA can be synthesized in the 5' to 3' direction. We all know that. Friends, at the end of the 5' there is a specialized region is present. What do you call that particular specialized region? You call it as UTR. What is UTR, friends? Untranslated region. What do you mean by the word untranslated region? This particular region not at all involved in the synthesis of protein, is it? So friends, this particular UTR, what do we call it as, friends? We call it as a leader sequence. What do we call? Leader. Now after UTR, what is present? There is a presence of the start codon. We all know very well, friends, the start codon is basically AUG methionine. Later what, friends? These are all codons. All are codons which are basically coding for the information. Okay, all are codons. Now friends, here comes one interesting point. What is that interesting point? If I ask you, friends, what is the length of the mRNA? How can you say that? Friends, length of the mRNA can be differ from based on the number of codons. If the number of codons that particular mRNA coding for, suppose example, I'll tell you one example. Suppose this mRNA coding for a protein insulin. It consists of how many amino acids? Just 51 only. Then this mRNA cannot be a too much length. Suppose example, it is going to code for a protein called as urease. It require more than 2 lakhs of amino acid. Then you only try to imagine how long the mRNA will be. So friends, the length of the mRNA can be varied based on the number of codons present. After the codons, there are 3 stop codons are present. We all commonly know that, friends. UAA, UAG, then friends followed by the UGA. These are all the 3 major stop codons that are involved in the, they are basically not coding for any kind of a amino acid. That's why we called them as what, friends? Stop codons. At the finally, at the 3' end, what is present, friends? At the 3' end, 3' end also there is another untranslated region is present. My friends, what do we call this particular untranslated region? This untranslated region, we will be calling it as a trailer. So the untranslated region present in the 5' direction we called a leader. The untranslated region present at the 3' direction we call it trailer. And there is a start codon and there are number of codons. Based on the number of codons, the length of mRNA will be varied and there are 3 major stop codon. Okay, that's fine. Then friends, how can I define a word polycistronic and monocistronic? Everyone has to understand, friends. Polycistronic means one mRNA can code for multiple proteins. Whereas monocistronic, the structure is same, there is no doubt about it. But what is monocistronic, friends? One mRNA can code for only one specific protein. My friends, what you people has to remember for the exam, monocistronic originally occur in the eukaryotes. We know, right, friends, we are complex organism. In our body, one gene, one protein, one gene, one protein. But in case of a prokaryotic organism, they are very simple or primitive organism. Then what we can say, one mRNA is going to code for the multiple proteins, right? I hope everyone understands the differences between the polycistronic mRNA and the monocistronic mRNA. You have to know this, friends. This polycistronic and monocistronic, even we are going to read in the translation also. Do not forget that. Okay, now here comes a very unique point. This is very important slide. I want every one of you to understand. Friends, first everyone answer my question. You are able to see this is the gene and this is the site where the process of transcription actually start. So what I can call this site? I can call this is the initiation site where the process of transcription is going to start. We all of us know very well please listen to me very carefully. If you understand carefully, by that you can understand the depth of the concept. You can answer any kind of a question easily. Now what I am saying, this is the site where the process of transcription actually start. But my friends, we all know very well, we're gonna read in the next slide, who is going to start the process of this particular transcription. We all know very well the versatile enzyme, that is the RNA polymerase enzyme. Friends, the point here is that, now every one of you listen to me. Now understand, do you think RNA polymerase directly come and attaches to this initiation site and start the process of transcription? No, this is absolutely wrong. Why? Because RNA polymerase enzyme do not associated with the promoter, do not associated with the initiation site directly. Then where exactly this particular RNA polymerase will come and attaches? Friends, this particular RNA polymerase will come and attaches to the specific site. What do we call that particular specific site, friends? We call that particular specific site only as a promoter. Never ever forget that. Now friends, how can I define the word promoter? Everyone listen the definition. Promoter nothing but what? It is a group or a sequence of nucleotide. Once again I am defining. It is a group or sequence of nucleotide through which, who will come and attaches, friends? Through which RNA polymerase will come and attaches. In a simple way, I can give you a definition, friends. RNA polymerase binding site. What, friends? RNA polymerase binding site. We will be calling it as what, friends? A promoter. RNA polymerase binding site, what do we call it as? A promoter. Never ever forget this particular point. So friends, first RNA polymerase enzyme comes to the promoter. From the promoter, it start moving to the initiation site. Then it will start the process of replication. Now you can ask me, how many promoters are there in the prokaryotic organism? And friends, how many promoters are there in the eukaryotic organism? Friends, every one of you has to remember that. You know, right? Bacteria or prokaryotic organism, very simple organism, my friends. Hence, there are very less number of promoters are there. But my friends, when you look into the eukaryotic organism, what is the point? Every one of you has to understand. In case of a eukaryotic organisms, friends, there are many number of promoters are present. In case of eukaryotes, that we're gonna read in the eukaryotic organism. In case of prokaryotes, scientists identified different type of promoters. One promoter, where it is located, friends? One promoter is located at -10 nucleotides. Now you can ask me, sir, why you are calling it as a minus? Why you have given the negative sign? Tell me. Because every one of you look into the picture very carefully. This is one of the fundamental difference. I want every one of you to remember for the exam. What is that difference? You are able to see, friends. This is what site? This is called as RNA transcription start site, which means from where the transcription is starting. So RNA polymerase enzyme will move like this. But friends, where the promoters are present opposite, downstream. What do we call? Downstream of the initiation site. Once again understand, the promoters are present on the downstream. So that's why what do we can call it them as, we will give it a negative sign because they are present down. So from here 1 2 3 4 5 6 7 8 9, so 10 nucleotides, there is one promoter is there. And what is the sequence of that particular promoter? Correct, Sonam. Promoter consists of TA, TA, TA. We can call it as a, originally we call Tata box. Even friends, we can also call it as a Pribno box. Very simple. And same, once again, if you start counting from the starting side 1 2 3 4 5 6 7 8 9 10 11 13 like that, if you count up to minus 35, once again there is another promoter is there. It is made up of TTGACA sequence. I'm requesting all of you friends, please remember these two promoters and their sequences in the prokaryotic organism without fail. Friends, actually all the scientists thought that in case, listen, actually all the scientists thought that in case of a prokaryotic organism, there are only two promoters are present, that is one at minus 10 and another one is at what friends, minus 35. But later two intelligent Indian scientists, Richard and Gauss, they have identified that even there is another promoter is located between minus 40 and minus 60. What they understood? They understood that there is another promoter is present at the minus 40 and minus 60. My friends, one of the unique point every one of you has to remember here is that every one of you has to listen. This minus 40 and minus 60 is not present everywhere. I mean to say in every single organism. Only few organisms this specialized promoters are located minus 40 and minus 60. But let me also tell you one more point. Actually this minus 40 and minus 60 do not have any key role in terms of initiation process. What is the reason? Because our RNA polymerase enzyme will come and attaches to the minus 35 and minus 30 only. Okay, now here is a question to all of you. Think and answer my question very carefully. First, our versatile enzyme, RNA polymerase enzyme. See, everyone listen, this is the start site of the initiation. From this particular site, the process of initiation has to be started. But friends, what is the point every one of you has to remember? Our RNA polymerase enzyme will come and attach us to minus 35 sequence. I can call this minus 35 region is the binding site for the RNA polymerase enzyme, which is absolutely true. There is no doubt about it. Now my question to all of you is that RNA polymerase enzyme associated with the minus 35, but surprisingly, you know friends, RNA polymerase enzyme never ever start breaking the DNA from here. Why it is not breaking the DNA from minus 35 region? Why the RNA polymerase enzyme breaking the DNA from the minus 10 region? What is the reason? Can anyone tell me? Originally it is associated with what? Minus 35. But it starts opening the double stranded DNA from the minus 10 region. What is the reason, friends? As all of us know, this particular minus 10 consists of what, friends? Thymine and adenine. We all know, right? Thymine and adenine consist of only 2 hydrogen bonds. So friends, there is no need to spend a great amount of energy. So what I can say, it is extremely easy to break. Is it, friends? Very good. TA rich. Savita, of course, that is true. It's a non-template region. No, it's not like a non-template region, dear. Okay, that also makes sense. But the original answer will be what? It is a AT rich region. AT rich means what, friends? It consists of only 2 hydrogen bonds. Then it can easily break. Very, very good. Very, very good. Now, friends, now let's look into our versatile enzyme. What is our versatile enzyme? RNA polymerase enzyme. Friends, listen every one of you very carefully. If you look into this particular RNA polymerase enzyme, RNA polymerase enzyme having, friends, it is not made up of a one subunit. We all know that, friends, RNA polymerase is basically composed of 6 subunits. Always remember. Okay, what are that particular 6 subunits? 2 alpha units. So alpha 1 and alpha 2 units, 1 omega, 1 beta, 1 beta prime. Now why I have written only 5 here? You know, the 6th one, we can call it as a sigma factor. Friends, I want every one of you to listen my statement carefully. These 5 together, 2 alpha units, omega, beta, beta prime, together called as a core enzyme. But friends, every one of you has to remember another unique point is that the core enzyme never ever be active. What, friends? The core enzyme never ever be active if it is not associated with a sigma factor. So what is the function of sigma? See, when the sigma factor associated with the core enzyme, what do we call this? Friends, we will be calling it as a holo enzyme. What we will be calling, friends? We will be calling it as a holo enzyme. Friends, what do you mean by the word holo? Holo means what? Complete. Holo means what? Complete enzyme. So now you can ask me a question. Sir, what is the major function of a sigma factor? As I told to all of you, these 5 subunits are having a core enzyme activity. So those 5 subunits only involved in the transcription, that is synthesis of the mRNA. Then what is the function of a sigma factor? Do you think without sigma factor the process of transcription will start or not? Without sigma factor, the process of transcription never ever starts. What's the reason behind that? Because I was continuously saying that RNA polymerase enzyme will come and attaches to the -35 region. That is what I told to you. Here RNA polymerase enzyme come and attaches. But the question is very clear. You know, right, DNA is a very long molecule. But in this entirely large DNA, how RNA polymerase recognize exactly where is the promoter, how it can recognize that much specifically? So that can be recognized with the help of what, friends? With the help of the sigma factor. Please kindly don't forget that point at all. Now see, don't get confused with the given statement. You have to remember, friends, sigma factor is exclusively important and responsible for or helping the RNA polymerase or guiding the RNA polymerase to recognize where exactly the promoter is located. A simple point. Another unique point what you people has to remember, friends, see, sigma factor is only required for initiation. What I mean to say, if sigma factor is continuously associated with the core enzyme, transcription never ever starts. What do you mean by that? Which means once the RNA polymerase enzyme recognize the promoter, immediately, my friends, the sigma factor has to be dissociated. What, friends? The sigma factor has to be dissociated. If sigma factor is present, RNA polymerase cannot move at all. So what is the conclusion here? Sigma factor is only responsible for recognition. It do not have any other key role. A simple point. Very good. Sigma 70. Now friends, everyone listen. When you do the cross section of an RNA polymerase enzyme, this is how RNA polymerase enzyme looks like. What are the various sub-compartments present in the RNA polymerase enzyme? Every one of you look into this picture very carefully, friends. You can see here there are different compartments are there. One we called it as what, friends? Look into this template strands channel. See, first of all, look into this channel, downstream DNA channel. What do you mean by the downstream DNA channel? Through that particular channel only the entire double-stranded DNA can be passed. That's why we call downstream channel. But as all of you know very well, we no need to use both the strands of the DNA. Not at all. We have to use only one strand of the DNA only. You all are aware about that. What is that particular strand we can call, friends? We call it as a template strand channel. What do we call? Template strand channel. And here, okay, through this is what do we call template strand channel. My friends, what is coming out? You are able to see, right? What is coming out? RNA is coming out. So that direction or the channel through which RNA comes out, we call, friends, RNA exit channel. Don't forget that. This many channels are there in the RNA exit channel. Friends, the point here, we have used only one strand of the DNA channel. Is it or not? Of course, yes. Then what about the other channel? Friends, everyone listen. What happens to this particular other strand? It will come out like this. This is called as a non-template strand channel. So downstream channel through which DNA will come, only one strand can be called utilized. We call template strand channel. One channel goes out. We can call non-template strand channel. RNA exit in the 5' to 3' direction. We call RNA exit channel. We all know very well. Even we read in the replication also. In which direction nucleotides will be added, friends? We all know very well. Nucleotides are added in the 3' hydroxyl group. Now this is what channel we will be calling it as, friends. We will be calling it as a nucleotide triphosphate uptake channel. Please kindly do not forget this particular statement at all. What do we call? Nucleotide triphosphate uptake channel. Don't forget the statement at all. So that is what, friends. Right now what we discussed till this particular slide is what are required for the process of transcription. We understand RNA polymerase, we understand sigma factor, we understand different subunits of the RNA polymerase, we understand what is transcription, we understand different type of RNAs, we understand the fundamental differences between the replication and transcription. Now we understand what are required. Now let's try to understand the process very clearly. The process of transcription. See, friends, we all commonly know that there are three common processes. Whether it is a replication, whether it is a transcription, whether it is a translation, doesn't matter. Basically it consists of what? Three processes: transcription, initiation, elongation, termination. Now there is a simple question to all of you. Before this particular initiation, there is another step is there. What do we call that particular step? Is recognition and binding. Recognition and binding. There is another step is there. Now my question to all of you is, what is going to be binded and what is going to be recognized? Which area is going to be recognized? Which is going to be bind? Yes, everyone. Which is going to be recognized and which is going to be bind? Anyone? Which is going to be recognized and which is going to be bind? Friends, very good. Most of you people know that our sigma factor is going to recognize what, friends? Promoter. Promoter to be recognized. Very good. And what has to be bind, friends? RNA polymerase enzyme has to bind. Very simple. RNA polymerase is going to bind, and who is going to recognize this? Promoter is going to be recognized, RNA polymerase is going to get bind. Okay, friends, now you look into this step by step. Initiation, elongation, termination. Friends, in this picture, I want to show you one simple thing. That is, you only able to see the sigma factor, friends. It is present only up to the initiation. Now you only try to understand this unique point. In the process of initiation, this particular sigma factor is present. Whenever the elongation will start, sigma factor get dissociated. So, and please kindly don't think friends, sigma factor should be present throughout the initiation process. Don't think that the entire initiation process contain the sigma factor. That is absolutely a wrong point. Listen my statement, I'll show you the animation. Sigma factor is exclusively responsible for what purpose? Just recognizing the promoter, then immediately it fall down. So after starting the initiation, sigma factor will be dissociated. In the process of elongation, in the process of termination, we do not require any kind of a sigma factor. Okay, now let's start one by one step elaboratively. So first step is what, friends? Initiation. Before initiation, just now even every one of you answered very clearly that there should be a binding. What has to be binded and what has to be recognized? Everyone of you look into the picture very carefully, friends. What has to be binded? This is what, friends? RNA polymerase. What enzyme? Core enzyme. This is what do we call? Sigma factor. All together, what do we call, my friends? All together we call it as a holo enzyme. Now who will come and attach us here? RNA polymerase with the help of a sigma factor. I have told to all of you very clearly where exactly it attaches, friends, at the -35. Okay, absolutely fine. RNA polymerase enzyme with the help of a sigma factor, it will come and attaches to the -35. Here comes another unique point. What is that? Does the RNA polymerase, upon recognizing the -35, does it starts opening this particular -35 region? Not at all. It never opens. You only look into this. Friends, this is a very important statement. Listen to me. This is, everyone look into this. This is the DNA. Okay. Now my hand is the RNA polymerase enzyme. Now you look into this, the edge you are able to see, right? This edge is the -35 sequence. This is the -10 sequence. You know, RNA polymerase will come and attaches very tightly here. Very tightly. When it attaches very tightly, this complex, we call it as what? Closed complex. My friends, please understand, use your brain. If RNA polymerase is present at the -35 region only, do you think it can open the double-stranded DNA? Not at all. If it wants to open the double-stranded DNA, RNA polymerase enzyme has to move. It has to move from where to where? It has to move from -35 region to the -10 region. Then whatever the closed complex which is situated like this closed complex has to be converted. What, my friends? This closed complex has to be converted into open complex. My friends, how is this possible? How the closed complex is going to be converted into the open complex? Every one of you listen to me very carefully, friends. Most of the people will think that in order to convert the closed complex into open complex, we require a great amount of energy. No, there is no requirement of energy. Then you can ask me, sir, then how this much tightly associated complex will become loose? Just because of a structural change. Just because of a structural change. You can also call it as what, friends? Conformational change. Conformational change or structural change between what to what? The structural change between my hand is RNA polymerase enzyme, between RNA polymerase enzyme and DNA, promoter present on the DNA. Because of the conformational change, because of the structural change, the closed complex can be converted into, friends, open complex. Okay, now that is what? Binding of the RNA polymerase, formation of a closed complex, then, my friends, formation of a open complex. Now every one of you listen to me very carefully. The first step, please ask me if you find any kind of a doubt. The first step, the chain initiation. Let me tell you one interesting point about the RNA polymerase enzyme. First of all, everyone look into the picture carefully here. This is what, my friends? RNA polymerase enzyme. This is the sigma factor. Where it has been come and attaches? It attaches to the -35 sequence. Then you only look into this. minus 35. Then where it starts opening the double stranded DNA? Minus 10. Now friends, every one of you just say yes or no to me. My question is, when the RNA polymerase enzyme open the double stranded DNA in the region of minus 10 promoter, my question is, does it start the synthesis of RNA from the minus 10 promoter site, or does it has to start the synthesis of RNA from the initiation site? Which site RNA polymerase enzyme has to synthesize the RNA? At the minus 10 region or at the initiation site? Everyone. Correct, Kushagree. It will help in converting into open complex. Correct. Can anyone tell me at which site the process of transcription will start? At the initiation site it is going to be started, or it is going to be started at the minus 10 region? Anyone? Come on, come on. We all know it. Obviously friends, our RNA has to start the process of transcription at the initiation site only. Correct, Harsha, that's true. No, 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 no. Everyone, whomever said minus 10 region, absolutely wrong it is. Friends, how it is minus 10, everyone look into this particular slide. People did not understand the concept. See here, this is minus 10. Now, where the transcription has to start, friends, from here. RNA polymerase should not start from here. You're getting my points or not? It should always start from a particular site. What do we call that particular site? We call the initiation site. Never ever forget. But why I am saying this point to all of you? Because friends, listen. RNA polymerase enzyme having a curious behavior. RNA polymerase enzyme having a very curious behavior. What is that curious behavior? It starts synthesizing the RNA molecule from minus 10 regions only. But once again you will get a doubt. Sir, if it is starting the minus 10, then why the initiation site is required? My friends, even though it starts the synthesis of RNA from the minus 10, it never ever synthesizes the RNA completely at all. Then what it is doing? You only look into the picture. It starts synthesizing the RNA in the form of a small pieces. So till when these pieces will be released, friends? These pieces will be released, pieces of RNA will be released till RNA polymerase cross the minus 10. Once the RNA polymerase reaches the initiation site, it start continuously synthesizing the RNA. Now you only try to look into the point. The moment RNA polymerase enzyme reaches the initiation site, sigma factor dissociate. Now the statement every one of, yes dear, in open complex only they are going to get separated. Correct, Sonam. What is the unique point every one of you has to remember? Sigma factor is only required for starting of the initiation. Sigma factor do not required for the continuation of initiation, elongation as well as the termination. So I am summarizing it quickly. Understand everyone who did not understand. Whenever RNA polymerase enzyme with the help of a sigma factor, it comes and attaches to this particular DNA. It is called closed complex. After the conformational change, it converted into an open complex. But RNA polymerase enzyme, before reaching the initiation site only, it starts synthesizing the RNA in a small pieces. This is, you know, what do we call, friends? Abortive initiation. I want every one of you has to remember this term, friends. Just now I told you na structural change dear. Okay, structural or conformational change. Deepika, look into the pen. This is the cap which is associated with the pen. You know, right? If the cap wants to fit into the pen, the cap should have a particular size, particular shape. At particular size and particular shape only it can fit into this. Now what I have done, I have changed the shape. Do you think this cap can fit into the pen? Of course it cannot. Then what we can say, upon structural or conformational changes, it lose the ability to fit into the pen. Same way, RNA polymerase enzyme and DNA associated like this. When there is a change occur in their structure, obviously they will convert it into a loose. That is called open form. You got it? Yeah, what we are talking, friends? We were talking now once the this initiation, what do we call abortive initiation? Why? RNA polymerase starts synthesizing these small pieces of RNA before reaching the initiation site only. Now, once it reaches the initiation site, it starts synthesizing the RNA continuously, and the sigma factor is also going to get dissociated. Sonam, just now I told you, no dear, see, if I want to open a cap and if I want to close a cap, it is not energy dependent. That's what I told you starting only. It's a structural change. Structural change is always not energy dependent. This structural change is energy independent. Do not require any energy. Got it? Yes. Next step is the elongation. Everyone commonly know the process of elongation, where the nucleotides are continuously adding. Now friends, every one of us understand Sonam, the most important and common point. What is that? We read in the first class also. In case of prokaryotes, transcription and translation are coupled. Yes or no? What do you mean by that? See here, mRNA coming out. In which direction mRNA coming out, my friends? 5' to 3' direction. I want every one of you listen, my friends. Even when 10 to 20 nucleotides of mRNA comes out. How many, friends? When 10 to 20 nucleotide of mRNA comes out, that is sufficient. Immediately, who will come and attaches to this particular mRNA? Ribosome will come and attaches. Ribosome will come and attaches. Immediately, what happening? The protein synthesis will start. That's why in case of a prokaryotic organism, that transcription and translation are coupled. This is the major reason. In case of a prokaryotic organism, there is a no post-transcriptional processes occur. There is a no post-transcriptional processes in case of a prokaryotic organism, because the process of transcription and the translation are highly coupled with each other. Means as soon as a little part of the mRNA comes out of the DNA, immediately what is going to happen? The ribosomes will come and start the synthesis of proteins. Never ever forget this point at all. Now you will get a doubt. Sir, how long this particular RNA polymerase enzyme is moving continuously? How long it can move? Until and unless it reach a particular site. What do we call that particular site? We call termination site. Now I will ask a simple question. I hope everyone will give the correct answer. My question is, where the termination site is present, friends? Does the termination site is present in the DNA or RNA? That is my question. Does the termination site is present in the DNA or RNA? Anyone? I did not ask any difficult question here. Yes? Where does this particular termination site is present? Does it is present in the DNA or in the RNA? Anyone? Yes, yes. Friends, the termination site is obviously present in the DNA, friends. Why it will be present on the RNA? You're getting my point or not? The point is very clear, friends. I am asking where the termination site is present. RNA polymerase enzyme, when it starts continuously moving, it has to stop at a particular point. The point at which RNA polymerase is stopped, that point only we call termination point. See, RNA polymerase function is what, friends? It is involved in the synthesis of RNA. Why the RNA having a termination point? Very good. RNA do not have a termination point. Always DNA having a termination point. Very good, friends. Perfect. Okay. Now when you look into the process of termination, there are two different process of terminations are there, friends. We will be calling it as a row independent termination, then a row dependent termination. Friends, sometimes instead of calling row independent and dependent, they can also call it as extrinsic and intrinsic termination. So this is what intrinsic termination means. RNA comes on its own out. RNA come out on its own. That's why it's called intrinsic. What about the extrinsic? RNA cannot come on its own. It always require a particular help. That's why we will be calling it as a extrinsic pathway. Kindly do not forget this point. Let me explain you how this is exactly happening. So first, let's understand row independent. What do you mean by the row independent? Without the protein row only, RNA polymerase and RNA coming out easily. Friends, everyone look into the picture very carefully. This is a RNA polymerase enzyme. Right. Now, you only try to understand this black color one is what, friends? This is what we can call the mRNA. So friends, I want every one of you has to remember one unique point. At the 5' end of the mRNA, please listen everyone. At the 5' of end of the mRNA, many GC sequences are present actually. Many GC sequences are present. Now, what is going to happen, friends? This particular GC sequence, we know, right? They can pair with each other. Upon combining or upon pairing of many GC sequence together, they can form a hairpin-like structure. Now, everyone has to listen my statement. Once the hairpin loop is formed, that hairpin loop pull the RNA. It pulls the RNA in such a way that RNA come out of the DNA. Here you are able to see, right? RNA and DNA are still connected. So we require a force to pull. So that force is created by whom? That force is actually created by this particular hairpin. So hairpin pulls the RNA in such a way that it breaks down from the DNA. Even with the help of with that force, RNA polymerase also terminate, or it also falls down from the DNA molecule. Here, do we use any kind of a external protein? No, we did not use any kind of a protein at all. That's why we called row independent. Without protein, we can do it. Without support. RNA without any one support, on its own it came out. That's why we call row independent. But you have to understand, if you ask me a question, sir, in our body, what kind of termination occur most commonly? You can get a question. In our body, mostly, friends, row dependent termination occur. Why? Because, friends, all the time, these hairpin loops are not strong enough. Once again, listen. All the time, these hairpin loops are not strong enough. That time, what we have to do, friends, we have to pull. So we require an external support to pull this particular hairpin loop like structure. Who is going to provide that force? That force can be provided by a protein called as a row protein. Now, please kindly don't confuse where this particular row protein will come and attaches to the mRNA. Friends, everyone listen to me. On the mRNA, everyone understand, on the mRNA, there is a specialized sequence is present. What do you call that sequence? You call the RUT sequence. Means row utilization terminus sequence. Once again, row utilization terminus sequence. So exactly here a row protein will come and attaches. Now you see here row protein. Here it was not showing. This is the hairpin. Hairpin will be present actually. Now the row protein will travel till the hairpin. What, friends? Row protein will travel till the hairpin. Then the row protein is giving an extra support for the hairpin to pull the RNA. Now with the help of a row protein, the RNA can easily comes out. This is what do we call row dependent termination. where always there is a requirement of a external protein row. Here there is a no requirement of a external protein. That's why called intrinsic termination. On its own it will come. Extrinsic termination always require the help. So friends, this is about the process of prokaryotic transcription. Of course, when compared to the eukaryotes, it's easy. Eukaryotic transcription is extremely difficult. Include a lot of promoters, include lot of factors. The process is difficult, but I'll make sure that I'll make it very simple in the tomorrow class. Friends, is that clear everyone so far? Everyone clear with the prokaryotic transcription process? Please say yes or no. Please give your comments if anyone having any kind of a doubt. Yes, you can say, Harsha. It's a kind of combination. Both G and C are present. G and C are present. Both are present. Both are present. Is that clear, friends, everyone? No. Kushagri, always remember, raw protein will give a support to the hairpin when the hairpin will become weak. So that's what I'm saying now. All the time hairpins are not stronger, they are weaker. So to support the hairpin, there is an external protein will come. Okay friends, that's all for now. Tomorrow we will meet at 7:30. We will finish the most important one, eukaryotic transcription. Okay, thank you all of you. I'll see you in the tomorrow class. Thank you. Good night. Please give your comments if you understand the concepts. Thank you all. Thank you. Bye bye. Good night.